Bismillah. Ya. Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Halo classmates dan sobat interested dimanapun berada, selamat datang. Bagaimana kabarnya? Semoga semuanya dalam keadaan baik dan sehat walafiat. Perkenalkan, nama saya Bea, saya dari Interest Education dan saya akan menjadi moderator hari ini. Interest Education merupakan konsultan pendidikan luar negeri dan juga tempat kelas persiapan IELTS dan TOEFL dan kelas bahasa Inggris lainnya. Teman-teman, terima kasih banyak sudah bergabung di acara pada Sabtu pagi ini dalam acara International Webinar The Future of Drug Discovery from a COVID-19 Perspective bersama Profesor Peter Cabot dari the University of Queensland, Australia. Nah, acara ini merupakan kerjasama antara kelas bersama kita, Interest Education, dan tentunya the University of Queensland, Australia. Dan acara ini juga didukung oleh Perpustakaan Kementerian Pendidikan dan Kebudayaan Republik Indonesia. Dan terima kasih kepada semua pihak yang telah mendukung acara ini. Baik, nah, teman-teman jangan lupa like dan subscribe channel YouTube Kelas Bersama Kita dan Interest Education. Nah, teman-teman, agenda kita hari ini ada 2 topik, sehingga selama 45 menit ke depan kita akan membahas mengenai the future of drug discovery from a COVID-19 perspective, dan Profesor Peter Cabot juga akan memperkenalkan program MPIP atau Master of Pharmaceutical Industry Practice School of Pharmacy di University of Queensland. Nah, di sesi terakhir akan ada sesi tanya-jawab selama 10-15 menitan. Nah, teman-teman selama acara berlangsung atau nanti ketika sesi tanya-jawab, teman-teman silahkan menuliskan pertanyaan teman-teman dalam bahasa Indonesia dulu, dan saya akan membantu menerjemahkan ke dalam bahasa Inggris untuk disampaikan kepada Profesor Peter Cabot, dan nanti saya akan coba juga membantu untuk merangkum jawaban beliau ke dalam bahasa Indonesia. Nah, teman-teman, sebelum ke inti acara, izinkan saya menyampaikan terlebih dahulu profil singkat Profesor Peter Cabot. Pertama, beliau adalah Direktur Program Master of Pharmaceutical Industry Practice di School of Pharmacy, University of Queensland. Beliau sudah bergabung di UQ dari tahun 1999, setelah sebelumnya menempuh postdoctoral fellowship di Johns Hopkins University, Amerika Serikat, dan juga pernah bekerja di the National Institute of Health Amerika Serikat. Nah, sudah 20 tahun ini beliau berkiprah sebagai dosen di bidang drug development and drug discovery, dan sekarang juga beliau merupakan peneliti praklinis di bidang analgesia dan ketergantungan opioid. Beliau telah meluluskan 20 mahasiswa dan mahasiswi peneliti alias yang S2 dan S3 by research, dan juga telah menerbitkan lebih dari 77 artikel ilmiah. Nah, karir beliau ini banyak berhubungan dengan riset-riset yang didanai oleh industri, dan sekarang ini banyak fokus pada mengembangkan dan meneliti opioid jenis baru untuk pengobatan rasa nyeri dengan potensi kecanduan yang lebih rendah. Baik, teman-teman, semoga teman-teman dapat menikmati dan mendapat banyak manfaat dari kegiatan hari ini. Sekarang mari kita undang Profesor Cabot untuk bergabung bersama kita. Untuk tim bisa tampilkan video Profesor Cabot. Hello, Professor.
Hello, Bea. Thank you. Hello, everybody.
Hello, thank you. Welcome to Indonesia. It's an honor as well as a pleasure to have you with us, although virtually. So I've already informed the audience of the agenda for today, and now I will hand it over to you. So classmates, please welcome Professor Cabot.
Thank you. Thank you so much, Bea, and thank you for the fantastic invitation to come and speak to you this morning. I'd like to give you some insights this morning into what we see as the future of drug discovery and how learning about drug discovery from the past is informing our step forward into the future. So I'll have the first slide, thanks. So we just wait for the slides to start and then we'll get underway. Great. So yes, the next slide, please. So we're just waiting for that to move forward. There we go. And there's me in the lab So I'm as B has mentioned, I'm an opioid pharmacologist. So I work in preclinical drug discovery and essentially for a career alongside my teaching, I actually develop new medicines in terms of looking for drugs that are less addictive as new opioid analgesics. And so I work with industry and have done for many years, even while I was at Johns Hopkins in the US and in my the earlier part of my career as a researcher, a young PhD student, which was a little while ago, I must say. So this is some of the work that one of my students put together that completed a PhD a couple of years ago and is now back home in Malaysia as an academic. Thank you. So the market itself, when we look at the scope of what we're talking about in terms of drugs and prescription medications across the world, we think of it, and I put it in US dollars because it's much easier for all of us to think of it in terms of US dollars. It's predicted to be something in the order of US $3 trillion by 2025. So it's a very large industry. And when we think about it globally, we were all in the same situation in terms of the impact of health on us, particularly in this current environment with the pandemic. But even without that, we all have aging population. There's an increasing prevalence of chronic illness. The costs of healthcare continue to increase. And the general populace tend to want more personalized medicines. They want therapies and they want treatment strategies that work for them. So in essence, this number is probably in some ways underestimated rather than an overestimate. So it's a big global market. And of course, with this means that there's a big global industry behind it. Next slide, please. So there we are. And then looking at the industry, I tend to think of it in terms of its three sectors. Now, it mightn't be apparent. A lot of us appreciate pharmaceuticals, which is down on the bottom right hand panel there. And then maybe you think more, particularly if you have somebody or know somebody that's undergone some cancer therapy, that you might be exposed to something called a biopharmaceutical, like the monoclonal antibody treatments that are around. And then, of course, our vaccines. But also medical devices as the top right-hand panel. The medical device is also something that's regulated, and it's also something that has to be unique and is often patented in order to protect the invention. And sometimes we actually don't need a new drug. In a lot of cases, we actually just need a better way of delivering it. And so nanopatch type vaccination programs, nanoparticle delivery systems are prime examples of developments in this area, and some of those are coming out of UQ as well. Next slide, please. So when we think about the scope of what this industry is, the size of this $3 trillion industry, it actually is mapped out in something like this diagram, which is the discovery process itself. You can see down in the green end is the preclinical drug discovery area, which is probably what we're going to focus a little bit on today. But you can see the value of the dollars translates through, and you can see the scope of the amount builds and builds and builds as you move along the arrow towards the red. These are estimates of the costs it takes to get a drug through the whole process successfully. And so when industry undertakes a process of drug discovery and walks them through these different phases, so you see from research and discovery through to preclinical GLP, across to clinical trials, which are mapped out in phases. They're phases around phase 1 and phase 2. They're about toxicology and safety, which you've probably heard a lot at the moment when we're talking about COVID vaccines all over the world. Some are in early phase 1. Some are just past animal studies in the preclinical development. And some are actually out there in phase 3, which I think the one in the UK is currently in that state. And of course, the UQ one is entering that stage. And this is where you start to get larger populous groups. You get a whole range of different components of the population to assess their safety and their true efficacy in a whole range of different parts of the population. Of course, then you also have another part which exists in the industry, which is this post-marketing surveillance or review processes. So even when a drug is out and being used in the public, we're still looking to make sure it's safe and that we haven't picked up some adverse events in a population group where it wasn't seen. So you can see this whole process costs a lot of money. So companies have to recoup these costs, and hence why the cost of medicines can be so expensive. The other thing is that this whole component is regulated by government bodies. So in the US, you may have heard of it, it's the FDA. In Australia, it's the Therapeutic Goods Administration Group. It's a similar approach to the FDA. And as I understand in Indonesia, it's the Badan POM that is like the Indonesian FDA. So these organisations are what's called the regulators, and they'll follow these medicines as they travel through this process from the green through to the red. So you have to make applications and you have to share data with these regulators in order them to be, for the new medicine to be approved or the new device. Next slide, please. So today I want to focus on, so it's probably an area of great interest to myself, but it's the research and discovery aspects and what we've learnt about history in some ways and how that informs us for facing the challenges in the future to meet the demands and the needs of an increasingly growing aware population around their own health and the use of their medicines. It's a big challenge for the whole health industry itself. Next slide, please. So then going forward, we often think about drug discovery, which is a generation of new molecules that interact with these sorts of pictures. You can see here a synapse, and it's undergoing a reaction where it's releasing a whole range of chemicals out to cause an action. And you think about us mysteriously considering how this molecular interaction might occur and working through novel mechanisms into discovery of how to interact with this process or control the balance. So often when things go wrong in terms of disease or infection or pain or any range of incidents, it's usually there's an imbalance in the system. And so interacting with that endogenous system or learning more about it enables us to be able to modulate that system more effectively. So a big part in understanding how to generate new medicines is actually understanding how the actual event occurs or how the disease process occurs. So what the underlying pharmacology is. Next slide, please. But then that doesn't mean that all the time we're thinking about is molecular mechanisms. Sometimes we need to think about reflection on the past and actually what has been and presented to us as a part of history. So traditional sources for medicines is actually not something that we've actually, going forward, will be dismissing at any time soon. We know that there's a lot to learn. There's a lot that we have learned, and there's a lot still to learn from traditional medicine. So in Australia, we have traditional bush medicines, particularly with the Aboriginal population. And some of the discoveries that they've made over time, we still haven't unravelled, and a whole range of different treatments. So, of course, as a scientist, that excites me because there's an opportunity to learn and understand and then explore exactly what the mechanism is that's going on. Now, because I come from an opioid pharmacology area, this talk has probably got a little bit of an opioid pharmacology essence to it. And so this plant here is Papavna somniferum, which is a widely grown plant, a beautiful purple flower. Many parts of the world have it. And of course, you might also know it as the opium poppy. Next slide, please. So a major problem with this class of agents, and of course we don't make morphine. So one of the major opioids that we get out of this poppy is morphine. And we know that during the Civil War, for example, in the US, that about a third of the population was addicted to opium tincture or tea. That they'd often use in their drinks, or they'd use to obviously relieve pain as well as create an essence of sleep and of rest and of peace. So with this addiction property that we see, that we know exists with opium, and of course exists also when you extract the compound morphine from that opium, there was actually efforts made very early on to actually solve the problem. And one example of those is a compound called heroin. And now heroin was so named because it was a savior. It was a savior for addiction. Because the only problem is that, yes, people's addiction for morphine was cured, but they actually ended up with an addiction for heroin. But here's a couple of examples of where early versions of pharmaceutical regulation, which didn't really exist, you could say, to any real degree. You can see the marketing. This is glycoheroin. So it was actually marketed for cough in this instance. And then you can see this picture, which was an advertising picture. They actually also used heroin drops for teething babies. So it's almost laughable when you think of that today. If somebody said to you, okay, my baby's got a teething, let's get some heroin and place it on their gums. It's something that you'd think was kind of ridiculous. But this was where we didn't have proper regulation of medicines. Of course, the concept of using opioids still exists today, and these opioids that come from the opium poppy are still used as clinical agents today. So we need to make sure we learn a lot from history. Next slide, please. So another example, just quickly, this is one, this is the meadowsweet plant on the left here with the beautiful yellow flowers. For centuries, a tea extract has been made from that. And on the cartoon sort of drawing on the right-hand side is a willow bark. And of course, this is the source of essentially of aspirin. Next slide, please. And so this also has an interesting history because this is a medicine which has been used for centuries, drinking the tea of a whole range of different plants. There's quite a number of plants that have the precursor for aspirin, which is salicylic acid. And of course, the chemists learn to acetylate salicylic acid and you end up with aspirin, which is acetylsalicylic acid, hence marketed here by Bayer on old labels or boxes that you see here and part of a collection we have in our own museum. So it's interesting to learn from history. Of course, aspirin is still used. It's a wonder drug. It's a lifesaver. It was nearly ruled out because we found with a lot of these non-steroidals that not used effectively can cause damage to the lining of the stomach. And of course, we've found ways to work around that. And one of those was not to design a new drug, but was actually to coat the aspirin so it didn't affect the stomach. So that's how the pharmaceutical industry reacted to it, because they know it is a wonderful drug and it saves people's lives. So it's backed and used more appropriately, but fantastic for treatment of fever as well as a whole range of other heart conditions. It's very useful for. Next slide, please. So then learning from history, there are other parts of drug discovery that in some ways this probably sounds a little ridiculous, but serendipity is always something that we need to rely on. If you put yourself in the right place as a drug discovery person, if you're doing the right sort of research, sometimes luck will have a play in how that turns out. And penicillin is one of the great stories of actually serendipity actually making a major part of the drug discovery process. Of course, Alexander Fleming here and Howard Florey, and I'm sure they're names that you've heard before and thinking about drug discovery. And of course, as the story goes, it was actually when Florey, they were in the pursuit of anti-infective agents. So we're looking for ways to kill bacteria to help with with disease and infection. And of course, Florey, well, as the story goes, Florey went away on holidays, I think to the south coast in England. And on his return, he discovered a mold had grown on one of the plates like he's showing here. And it's just like one of those agar plates that you've probably seen if you've done a science degree or some microbiology at school. And he noticed that the mold that had grown there had a kill ring around it. In other words, the bacteria that he'd plated on those plates that he'd accidentally left had grown this mold, and there was this ring of bacterial death around that mold. And so, and anybody else, you, me included, maybe we'd throw the plate out and think, oh, that's disgusting, that's awful. It's like finding something moldy in your fridge. You wouldn't think, hey, this might be actually a great discovery here. And of course it was. The fungus was producing a compound which would become an antibiotic, which is known as penicillin. And of course, Nobel laureates follow for those sorts of discoveries. And of course, now we need to find more of these, because we know that with bacterial resistance, the pursuit of new antibiotics is something that's increasingly urgent and increasingly going to be a major issue for us going forward with the amount of use of antibiotics. So that's why you're probably hearing a lot on the news about reducing the use of antibiotics. This really was actually a problem that was generated as the penicillin was produced heavily for the war. They actually had too much of it in stockpiles. And so the company promoted it so that its use became expansive. And I think from that point on, we've never really controlled it effectively. So hence why we're in a state where we need urgent antibiotics. So not only COVID vaccines, but new antibiotics. Thank you. Next slide. So that's serendipity. The next part is something that probably at the turn of the century, around that time of the discovery of penicillin, we're also invested in scientific development or discovery based on a disease. So understanding a disease and its process. And one of the greatest examples of that is diabetes. I talked a little bit this at the start. And so the more we actually understand how the disease is occurring and what's happening, the molecular mechanisms that underlie it, the ability for us to be able to understand how the body manages it or what's wrong with the balance becomes more aware to us. And diabetes is a great example of this, where it was discovered prior to the research by Banting and Best here. The pictures are shown that, so Paul Langerhans described that the pancreas cells, or the Langerhans cells, which are named after himself, I guess you got to do that in those days, he understood that this was an important part of the process of diabetes, because diabetes at this point, so this is type 1 diabetes, this is, or you might hear it termed insulin-dependent diabetes. For those of you that have this, you know this all too well, you're required to take insulin. So the strategy actually hasn't changed heavily over time, but we know that it has to do with an imbalance in insulin. Now, they undertook this process of thinking, well, how do we prove this? So, which will sound a little harsh, but what they actually did was they removed the pancreas from, like this puppy here, which is called Majori, and then they fed back beef and pork extracted homogenized pancreas. And in doing that experiment, they learnt that it truly was something that was in the pancreas that was actually generating the capacity to control the blood sugar levels and actually inhibit the diabetic issue. So this process of feeding back that extract allowed them to then go and isolate it in this lab here, the extracts from the porcine and pig, so the pig and beef pancreas, and able to interpret exactly what was going on in terms of a treatment strategy. Of course, again, this was a Nobel laureate from what seems a simple experiment. However, this was them understanding the problem more, allowed them to actually generate a better perspective on a treatment strategy, because we don't use pig and cow insulin anymore. We actually have small cells that are recombinantly produce insulin, which is human, and that will be often used in products called Humulin, I think it's actually called. I'm not sure if that's the one that's labelled in Indonesia, but certainly that's the most common form of insulin. So next slide, please. So then, okay, some chemicals. So we all like good chemical structure in the morning. On the right-hand side is actually the prototypical compound called morphine, as I always default to opioid analgesics. I apologise for that. On the left-hand side is a open chain analgesic like fentanyl. So fentanyl is a very potent analgesic and more clinically used than morphine these days. And that's because it's very selective and very potent as well. So it makes it economical, but it also is a very good clinical agent. Now, as a drug scientist, we can look at the structures here and determine where the points of activity are. So on the right-hand side, morphine, as you saw it there, you can see the red oxygens, but you can see the blue nitrogen. And that blue nitrogen is actually the key point in terms of what we call the pharmacophore, or the active structure within that molecule that predicts it to be an opioid. So in science, we now use molecular modeling to tell us where these pharmacophores are in compounds and allow us to predict the activity of the drug. Now, on with the fentanyl compound over here, the bottom nitrogen in blue there is almost the same type of pharmacophore behavior as the morphine one on the right. So as a scientist, I'm aware of that. And so in the design of these compounds, we know that we can build around this pharmacophore structure and still maintain an opioid type activity. So next slide, please. So then when we look at a group of opioids, we get an interesting story. So these all look pretty much the same, and they are all, you can see the names are all very similar: fentanyl, sufentanil, and alfentanil. Now, these are all clinically used agents, depends on the part of the world you're in, because different regulators take on different drugs. Fentanyl is more commonly used in Australia for pain relief. But you can see the nitrogen conserved components within those virtually in the center of the picture. And that's what we call the pharmacophore. So in combinatorial chemistry, we learn about that pharmacophore and we build the structures around it. But what this can amount to is hundreds of thousands of structures, as you could appreciate, a high volume of these structures that all have to be tested. And now up in the top right-hand side is one that actually was too potent. It's actually 1,000 times more potent than fentanyl. And it's so that you'd have to think fentanyl is more potent than morphine. I think carfentanil ends up around about 10,000 times more potent than morphine. So that is a very dangerous drug and often thought of as a war chemical because it's so potent. Next slide, please. So very quickly, a bit of an example of this, and this is where an opioid discovery program ends up with a chemical that's not a very good chemical for use in humans. It's actually a very effective chemical for use in sedating large animals like elephants or rhinos for veterinary purposes. But it's actually also been classified as a war chemical in some parts of the world. And here's an example of a paper that looked at clothing and urine from deceased peoples during a Moscow theatre siege some time ago. And if you recall it wasn't all that long ago, but probably a decade ago. And this is where a chemical was deployed into or a mist of a chemical was employed into a theatre to rescue hostages. In essence, they used carfentanil, but it was too much. And so a large number of people died. You can see 125 people died. And so one of the adverse effects of opioids is respiratory depression. So usually you can reverse it with a compound called naloxone, but you have to be very quick to get that too. So, which is used in ambulances. So for people who have heroin overdoses, I don't know how effective naloxone is against reversing carfentanil. But of course, this is an illicit drug in the open markets as well, or on the black markets, I guess you call them. Next slide, please. So I guess this is probably coming to where I'm at the moment. In terms, we don't tend to do combinatorial chemistry like that anymore as much. Now we actually have the capacity to do molecular modeling. And this is where we start to think about these new vaccines as well. Now, in the case of what I do, this is an example. The top left-hand panel is an example of DMT tick pharmacophore. There's a lot of language on this plate that we don't really need to explain, but the left-hand panel is essentially what we've drawn as a delta opioid agonist. So it works at a different type of opioid receptor than the morphine does itself, or fentanyl. And we've designed this in 3-dimensional. So this compound is not made. And so on the right-hand side is an example of the crystallized form of a receptor. So scientifically, we've been able to progress now that we can actually look at the crystal structure of a receptor where a drug works. We're then be able to draw a molecular model of this. Now, this is not my work, but this is the work of those in the public. But I do this sort of work in the lab that's right behind me. And what we can actually do is take that compound on the left and model it how it interacts with this receptor on the inside of the pore. And that's down on the left-hand picture down here. So this is very similar to what we do. And you can see the balls. It looks like one of those play pools where the kids have got all the balls and you throw them into it. But it's actually a molecular model of the inside of that receptor that's drawn there on the right. And so that is a G protein coupled receptor, the delta receptor, and down inside that, you can see it looks like that drug molecule. And so there's a chemical form of the drug molecule shown there in the middle next to that. But in 3 dimensions, it actually looks like that panel up there on the left. So we can actually, in vitro, in a molecular, so on a computer, actually model the interaction of that new drug that we've never made with a digital form of the receptor, and we can actually measure that interaction and determine our possibility for how that might interact and what changes that might cause to that receptor. What that allows us to do is allows us to make lots and lots of virtual compounds, screen lots of them with many receptors, and determine which ones will work, which ones might not work. And then from that point, so this, in the last 2 years with a company contract I've got with a company in Canada, we've been doing this for maybe 100, 200 chemicals and screening them through this type of virtual, and then this virtual process. And then we've made probably about 20 of these types of molecules, and then actually tested only those 20 because we figured out that they work in this molecular modeling. So it allows us to advance our drug discovery programs very, very quickly. It means that we don't have to be in animal models as early. It means that we don't have to be in cell pharmacology experiments as early. We can actually determine the right structure in a model. It all depends on how good the model is, though. Next slide, please. Of course, the other approach that we still use as well is we look to nature. And I think that's something that we've learned in drug discovery is you can't not consider what the natural environment is doing it. We live in that natural environment, we are part of it. So things that work in that environment also work on us and have the potential to be new medicines. So this is a program I've been working with some colleagues, sort of ancillary, but the center of the team is at an institute at UQ. And so this was a discovery process that team made, where they know that these cone snails, so you've heard of the Great Barrier Reef in Australia, and I'm sure there's cone snails around in Indonesia as well. And if you know anything of them, they're beautiful little snails. They move very slowly, but they have a deadly barb of poison. And this is an example, a molecular structure of a poison right here. And so what they do is when they are chasing their prey, which is very slow, they're a snail, but they have a very quite subtle way of evoking or catching their prey. And they send this barb out, and what it has in it is it has a toxin in it that means that when it stings, you actually can't feel the bite. So it's fairly unique. So when scientists understood more about this, they thought, well, hang on a second. This means there must be some type of analgesic or anesthetic capacity. So these conotoxins have a potential for analgesic or anesthetic capacity. So what they did was they took heaps and heaps of extracts of these venoms and put them through a high screening process to look for which ones make the greatest response on a certain type of channel, on a certain type of potential channel involved in pain. And so that allowed them to use high throughput screening to identify potentially a compound in nature that might actually help in terms of curing pain. Next slide, please. So then we think about things like vaccines. So, of course, prior to the COVID event, the cervical cancer, an insidious disease, in the institute that's next door to me here, next door to Pace, Professor Ian Fraser undertook the discovery of Gardasil, such a long program, and as a vaccine. And of course, that was a famous outcome for the world in terms of development of a new novel vaccine against a cancer. So, and not only in females, but also in men is it used today. And of course, there are a number of these vaccines now. And now vaccines, and probably from that point, vaccines have really risen as the new potential savior for a lot of conditions that we are afflicted by. Next slide, please. So then when we talk about our vaccine, the other vaccine that we have, which I think everybody's talking about vaccines, I think at the moment there's around about 150, 160 potential vaccines out there. We know the one in Oxford is in quite advanced stages. And of course, this is one that is closer to home and is just across the river from me here at Pace. And that's with the COVID-19 research team at UQ. Next slide, please. The interesting thing with this vaccine is it's a patented technology and it's part of a collaboration with the World Health Organization. And they've actually been exploring for a number of years the idea of treating a lot of serious diseases such as Ebola, influenza, which is a big killer, maybe not as, or definitely not as infectious and severe as SARS. The first SARS epidemic, of course, as well as this current SARS. And this is with a technology called a molecular clamp. And essentially what they've done in that is you actually don't need the live virus. So safety is a great aspect around this because they generate this peptide frame that they basically clamp from understanding the genetics of the virus itself. So it actually has no live virus content, but it's actually got the message component by which the immune system sees this virus. Oh, it's 5 minutes? Okay, thanks. So next slide, please. And then, of course, these, like all vaccines, these vaccines are generated to spike an immune response without providing any of the live virus behind them. So you stimulate, or you can often use an adjunct, which creates a danger signal, and then you have the antigen. So you're making the something that pretends to be the virus, and the body responds to it, develops antibodies, and then, of course, you have an endogenous immune system takes control and generates an immune response. And this is where it can be very different, because if a patient doesn't have a very good immune system response, they cannot generate the antibodies, so you can't protect yourself. So that's why the strength of the vaccine is really important, and what kind of immune response it actually generates. Next slide, please. So then back to our drug discovery life cycle. What type of roles fit across this process? Next slide, please. So not only do you have the drug researchers like myself, but you have a whole range of different careers. The next slide. It's a big slide, so it's probably hard to move. And so you can actually then see a whole range of different careers across this spectrum. These could be medical affairs, these could be medical writers, they could be marketing, manufacturing, health economists, quality assurances in manufacturing, as well as compliance officers. So the scope across this big industry is actually quite dynamic. Next slide, please. So to cater for this size of a career, or this scope of careers, we actually have generated a new type of master's program called the Master of Pharmaceutical Industry Practice. And that has a graduate certificate component as well. Next slide, please. So in essence, this is a 2-year full-time program. So I actually am along with my team here at Pharmacy, we've built this program, and we built it with the scope of the first year is focused on understanding that whole drug life cycle, understanding all of the aspects around taking a medicine from discovery, like I've talked about today, all the way through into post-marketing surveillance. Then in the second year, it provides the opportunity of 1 or 2 in-depth placement or engagement in research, and the research like I've talked about today that we're doing out here in the lab, or in health economics. Next slide, please. Of course, this program actually also it starts either in first semester in February or in July. And of course, it's here where I am right now today talking to you from, which is in our brand new PACE building. Next slide, please. And oh, sorry, that's another pictures. It looks like in summer with the storm coming. Next slide, please. Our lovely library, which won an award. It's a beautiful space. I call these booths the cone of silence. They're like little booths with leather lounges where the students sit. And then, of course, these are the labs where you undertake the laboratory component in the pharmaceutical analysis aspects that I teach. Next slide, please. And of course, this is the scholarship offering also that's available, but you can see more information on any of these aspects at the Future Students website at UQ. Next slide, please. And those are the links there. And thank you so much for the opportunity to talk to you today. I think the next slide, and we're open to questions. Thanks, everybody. Hi, Bea.
Hi, Professor. Thank you very much for a wonderful talk. Thank you. And also for the explanation about the Master of Pharmaceutical Industry Practice at UQ. So for those who are interested, you can contact us for more information on how to apply to that particular program. So now there's been a lot of questions coming in.
Oh, great.
Yes, but due to the time constraints, we'll be selecting 3 questions for us to discuss for today. If that's okay with you. So
Yeah,
the first
definitely.
question is, yes, thank you, is from Rizal Matondang, and he's asking, is there any plant medicines that can hinder the growth of COVID-19? And if there is any, what are the chemical compounds that are contained in those kinds of plants?
Yeah, I know there's a lot of, I guess one of the predicaments we have with COVID-19 is there are a lot of stories coming out around different things that can help or hinder or and we've seen a lot of it in the news, and some of it coming out of the US. I don't, from what I've read in the literature, there's nothing in terms of natural products that have been useful in the treatment of COVID-19. There are a number of compounds that have been speculated that might have be able to help in how the immune system handles the virus. However, I think the problem is with this virus is it took hold so quickly. Nobody's able to do proper scientific research to really figure out if it's truly been helpful. So I think at this stage, it's probably way too preliminary to say that there's anything. And I think that's why we need to probably focus mostly on the idea of a vaccine, because I think that's our only way out of this pandemic.
Yes, great. Thank you, Professor, for the answer. Yes, please allow me to just summarize a little bit in Bahasa Indonesia.
Yeah, definitely.
Yes, thank you. Jadi teman-teman, kalau menurut Profesor Cabot, belum ada kalau dari misalnya dari tanaman gitu ya, itu belum ada, dan karena COVID ini sangat cepat gitu ya kejadiannya, jadi belum bisa ada penelitian yang bisa dilakukan dengan benar-benar proper gitu ya, benar-benar teliti gitu ya untuk meneliti apakah misalnya produk-produk yang alami itu benar-benar ada efeknya begitu. Kira-kira seperti itu. Terima kasih, and we'll move on to the second question. I think it's from, sorry, so while we wait, I will try to, look at the question. Oh, sorry, it's from Riak Kencana. So what are the essential challenges for scientists to discover the vaccine of COVID-19? And are there any, what's the traditional substance to help the issues in discovering drugs for COVID-19?
I think the most important part, so the vaccine technologies have changed a lot. And I think the main step forward was actually getting the genetic code out from the vaccine very early, which was really fantastic and very helpful for scientists all over the world. Other approaches have been accessing the live virus. Of course, the predicament with these viruses, so it's an RNA virus, they are very difficult to develop vaccines for. But thankfully with some of the new technologies, like the one we talked about at UQ with the molecular clamp, you're able to actually adjust to that challenge very, very quickly. I think the difficult thing is you see the timeline when I talked about drug discovery process, and there it said 15 years. Now, if you took 15 years to generate these vaccines, we wouldn't have to worry about that virus anymore. It would be something else by then, particularly when you think about a virus like influenza that comes through different all the time. So the biggest challenge for vaccine development was actually getting something that works quickly, getting something that we can get out there very quickly, but also be able to show it's safe. And
Great.
so that's probably the two big challenges.
Yes, great. Thank you very much for that answer, Professor. Well, we wish you all the best in trying to discover the vaccine as soon as possible, so then it can be used by many people as soon as possible as well. So please allow me to summarize your answer. Jadi teman-teman untuk berdasarkan jawaban beliau secara singkatnya ada dua challenges ya, ada dua tantangan utama. Yang pertama gimana caranya untuk bisa mengekstrak kode genetik dari COVID-19 ini secepat mungkin gitu kan, terutama pas di awal-awal. Kemudian juga bagaimana membuat proses ini bisa lebih cepat lagi gitu. Tapi karena sekarang udah ada teknologi baru gitu ya yang mereka gunakan di lab, jadi ya prosesnya sekarang bisa jauh lebih cepat gitu. Tapi ya dua tantangan utamanya ya seperti itu untuk mengekstrak kode genetiknya dan juga bagaimana caranya agar pengolahan vaksin ini bisa sesegera mungkin, bisa secepat mungkin, tapi juga sekaligus aman gitu ya, aman untuk nantinya digunakan. Jadi tidak asal-asalan seperti itu. All right, thank you, Professor. We'll move on to the third question from Jehan Kapitan Hitu. How does the Queensland government or authority is dealing or managing the COVID-19 situation?
Yeah, that's a great question. So actually, they've been doing a really good job. I think Australia in itself is doing a really good job. The problem is, I guess, for everybody across the world is we're only learning about this virus as it's spreading. So so dealing with how the virus is working and understanding how it's infecting people, are actually having to learn while everybody's getting infected. So Australia took on what's called a suppression type model, and Queensland, as one of the states, has worked along that model as well, which is actually using the principle of contact tracing. So in other words, not trying to eliminate the virus, because we know that's probably impossible. And many countries have tried that process, and it doesn't tend to work completely, but suppress it. So in other words, when there is a hotspot or an outbreak, the people that are being in contact with that person are identified, and then typically they have to go into lockdown for 14 days and be tested throughout that period. And that's worked pretty well at this stage. We don't have a lot of cases, although we have had some outbreak in the last couple of weeks, unfortunately, but it's a very difficult virus.
Yes, it is. Thank you, Professor, for your answer. It's good to hear that there's been not that many cases again in Queensland. So please allow me to summarize that again for the audience. Ya, jadi classmates, kalau untuk saat ini salah satu hal utama yang dilakukan oleh pemerintah Queensland adalah melakukan yang namanya suppression type model, dan itu intinya adalah untuk menekan jumlah kasus melalui cara contact tracing. Jadi begitu ada hotspot, ada semacam zona merah, begitu semua orang yang berada di zona tersebut langsung dikontak dan juga langsung dilakukan contact tracing, dan juga langsung dihimbau gitu ya, di-encourage untuk melakukan karantina atau isolation begitu. Dan sejauh ini cara seperti ini sudah cukup efektif menekan jumlah kasus COVID-19 di state of Queensland khususnya. Thank you, Professor. We'll move on to the next question. So, Mas Giri, silahkan boleh ditampilkan lagi pertanyaan berikutnya. All right, so this one is from Muhammad Kais. All right, so well, it's actually about the COVID-19 in Indonesia, but it's about the mutation of the virus. Is it possible that the virus is stronger, especially because there are some other similar viruses of corona, right, in the corona family? And is there any other way to prevent or to overcome it besides the use of vaccine?
Yes, that's a great question. Whether there are drugs that will work to treat people once they're infected is still something that's being researched at this stage now. I know that, and you will have seen that yourself in the media and literature reports and papers, as well as there are a number of drugs that might be useful without the vaccine to actually treat it once a person's been infected. That still hasn't been completely scientifically validated in a lot of those cases, so there's still some research to be done. The predicament we've got is the virus spreads that quickly and changes as well. And we know that the part of the genetic tracing, we see the genetic code is slightly different in different groups of people. So these viruses do change. They're RNA viruses. And like you said, there's a coronavirus family. This particular group does mutate, but they tend to, and I guess this could be, who knows what the real answer is, but they tend to only change a bit, but who knows what the future holds with these viruses. I think we're learning a lot, but we're also learning that I think it's a vaccine is really our only way to be able to get a good block on this virus across a big population. So I don't see us being able to use a drug very soon, given that regulatory process I showed you with 15 years. So you can fast track viruses so they're different to drugs. The safety and efficacy proving stages are actually quite different. So we can actually take vaccines through the safety and efficacy stages much more quickly than drugs, because in a lot of cases, these vaccines don't use live virus. And so their safety then is a lot better than maybe vaccines that you've heard about used a long time ago.
I see. Great. Thank you very much, Professor. Allow me to summarize that again. Jadi classmates, menurut beliau sih kalau misalnya sejauh ini belum ada cara lain gitu ya selain kita menemukan vaksin, dan malah proses penemuan vaksin bisa jadi prosesnya lebih cepat daripada penemuan obat gitu untuk COVID-19 gitu. Dan lagi-lagi karena kejadian COVID-19 ini sangat cepat, jadi untuk penelitian-penelitian yang benar-benar lengkap gitu ya atau proper mengenai penemuan atau pencegahan COVID-19 ini memang masih belum bisa dipastikan betul-betul gitu ya. Jadi ya sejauh ini kesimpulannya adalah mungkin satu-satunya cara masih penemuan vaksin ini begitu. Jadi kita doakan semoga bisa segera ditemukan. Great, thank you, Professor. We'll move on to the next.
Sorry for such a long answer.
No worries. Yeah, I already got the main points, so I just I mentioned those main points for the audience. Great, so thank you. So we have time. Kita masih ada waktu for just one more question, if there's any more. From Rivaldi Pandi, how does this virus avoid our immune system? So, because isn't it easier for us to overcome this virus because this virus isn't really different from the normal coronavirus? I mean, what does research say about this, if there's any?
Yeah. So, thanks, Rivaldi. So if what you're saying, what are the differences between the viruses? I think that's something we learn. So a virus is part of an ecosystem, and I guess our traditional, a lot of coronaviruses we know from the past that exist in a lot of animals, such as bats and those sorts of animals. The transferability from animal to human is rare, and often it can be easily controlled. An infection outbreak is actually easily controlled. And we saw that particularly with the first SARS virus. It didn't really cause the pandemic. It had an epidemic status in some countries, but didn't create the pandemic. Of course, it was very, very deadly. So this version seems to have the capacity to spread a lot better in air. And so this is actually part of a difference in the virus design between the two different types of corona. And so that capacity to be able to spread more easily in the air then makes it even more dangerous, because it's much more easily transferred, and hence why we have the social distancing approach to controlling it. there's also then some evidence around maybe this will, because it does spread by air so well, maybe it is different in different climates. And so there are more things that we will come to learn about this virus as we study it further and further. But I think that the differences in essence can be quite extensive, even though they have the same name.
Yes. Yes, thank you, Professor, for your answer. Allow me to summarize that again. Jadi teman-teman, ya memang lagi-lagi kejadiannya untuk COVID-19 ini sangat cepat dan masih banyak hal yang belum diketahui dan masih terus diteliti oleh para peneliti mengenai COVID-19 ini. Tapi pada dasarnya perancangan atau desain dari virus yang COVID-19 ini khususnya memang berbeda aja gitu ya dengan virus-virus yang coronavirus yang lainnya begitu, dan dia memiliki, jadi dia COVID-19 ini memang memiliki kemampuan untuk menyebar lewat udara dengan lebih mudah gitu ya, atau lebih cepat gitu. Jadi sehingga itulah yang membuat kita kesulitan gitu ya untuk mungkin menangani atau mencegah dengan cepat gitu ya untuk situasinya begitu. I'm sorry, Professor, can I just confirm? So when you said it spreads easily through air, so it's, in other words, it's airborne. Is that?
Yeah.
Is that?
So I guess at the early stages of when the coronavirus hit, particularly in Australia, communication was around it, living a long time on surfaces and contact with surfaces. Now that still exists, but as we see more with it, the respiratory, so people coughing or sneezing, that airborne droplet type approach means that that it is infecting people through not just contact, but also through the air, hence why staying apart is an important part. So as we learn more about the virus, we understand more how we have to protect ourselves. And so that's where the strategy of the masks have come into play. Because if you're sick and you're wearing a mask, you'll expirate into that mask. And so you'll be protecting the others around you.
Yes. Great, thank you very much, Professor, for confirming that. Jadi teman-teman semua, memang makanya pentingnya menggunakan masker gitu ya, dan juga physical distancing karena melalui droplets gitu ya dari melalui droplets, dan begitu cepatnya virus ini menyebar melalui udara, jadi sehingga kalau pakai masker dan physical distancing akan sangat membantu gitu ya, dalam hal mencegah penyebaran yang lebih luas begitu teman-teman. Alright, Professor, ini kita sudah sampai ya teman-teman di penghujung acara. So Professor, thank you very much again for the wonderful talk and joining us for today. And thank you also for your valuable contributions. We hope to see you again, and please allow me now to conclude the event.
Right, thank you, Pia, and thank you for inviting me to your classroom today.
Yes, thank you very much. Jadi classmates semua, kita sudah sampai di penghujung acara. Terima kasih banyak atas partisipasi aktif teman-teman semua dan juga atas pertanyaan-pertanyaannya. Semoga bermanfaat. Ikuti terus kegiatan-kegiatan dari kelas bersama kita dan juga kegiatan-kegiatan dari Interest Education. Sedikit informasi bagi teman-teman yang berminat untuk kuliah di luar negeri, khususnya yang berminat untuk ke University of Queensland, seperti program yang tadi dijelaskan, bisa hubungi Interest Education. Mungkin gampangnya lewat Instagram di direct message, dan juga kalau misalnya perlu kelas persiapan bahasa Inggris seperti IELTS, TOEFL, dan lainnya bisa juga menghubungi kami. Untuk terakhir ini, saya ucapkan terima kasih banyak sekali lagi untuk teman-teman semuanya. Semoga bermanfaat. Sampai ketemu lagi di acara-acara selanjutnya. Wassalamualaikum warahmatullahi wabarakatuh.